In biological night vision, the rhodopsin in the rods of the eye breaks as light hits it. The peak rhodopsin buildup time for optimal night vision in humans is 30 minutes. As light enters the eye, it goes through the cornea, then through the pupil, and finally through the lens. During this entry, it is refracted or bent and then focused by the lens. Once this occurs, an image of what you see in front of you is burned, so to speak, on the retina. The retina is a thin layer of specialized cells located at the back of your eye that help to transmit information provided by the light to our brains. In order to recognize what we see in front of us, our retina has several different layers. One of the most important layers is made up of highly specialized neurons called photoreceptors that convert information provided by light into electrical signals that are conducted to our brains. These photoreceptors are called the rods and cones. Rods are called rods because under the microscope they are shaped like, of all things, a rod. These rods are located mainly toward the side of your retina. To sum this all up one more time, rods are photoreceptors located near the edge of the retina that are responsible for peripheral and night vision. Conversely, cones are photoreceptors that are centered in and on a depression near the center of your retina called the fovea, or more technically referred to as the fovea centrioles. Again, cones are photoreceptors located near the center of your retina that are responsible for your ability to see during the day, in color, and also in detail. Also, we have a series of light-sensitive cells inside of our eyes. Some of them get burned out when looking at bright light, but they quickly heal when no longer exposed to light. The rhodopsin receptor in the site where physics meets biology, where a photon of light from physical world must get interpreted for the nervous system. And this was said by Marie Burns, who is a professor of vision science at UC Davis School of Medicine and lead author of the study. Burns and her colleagues studied rod cells in the laboratory and discovered that calcium plays the gatekeeper role. They found that rhodopsin activity changed calcium levels in the cells and that overactive rhodopsins changed calcium levels at a faster rate than normal. This faster change led calcium to trigger a series of chemical steps to counter the overactive rhodopsin signal by producing an equal and opposite signal, thereby correcting false information before it gets sent on to the rest of the visual system. They uncovered this fundamental new level of control by measuring how long individual rhodopsin receptors remained active in response to flashes of light, and then determining how much calcium's gatekeeping function modified the rhodopsin signals.
So a rod is a photoreceptor. What exactly is a photoreceptor? Is it a neuron? Is it a type of nerve? So in fact, it is. It's a very specialized type of nerve that's able to take in light and convert it into a neural impulse. So inside a rod, there are a whole bunch of structures known as optic discs. And these optic discs are large membrane-bound structures inside the rods, and there are thousands of them in an individual rod. Embedded within the membrane of each optic disc is a whole bunch of proteins. And these proteins actually absorb light and begins the phototransduction cascade that eventually leads this rod to fire an action potential that will reach the brain. Similarly, a cone gets its name because it's cone-shaped. Cones are also photoreceptors, so they're specialized nerves that have the same internal structure as a rod. So cones also have a whole bunch of these optic discs that are stacked upon one another. And embedded within each optic disc is a whole bunch of this protein. So as I mentioned over here, the protein in a rod is known as rhodopsin. In cones, it's basically the same protein, but it just has another name, and it's called photopsin. So as I mentioned, as a ray of light enters the eye, if it happens to hit a rod and it happens to hit rhodopsin, it'll actually trigger the phototransduction cascade that results in this rod firing an action potential. This exact same process happens in a cone. So these are the major similarities between rods and cones. Now let's look at the differences. So in an average retina, there are about 120 million rods. In contrast, there are about 6 million cones per retina. So there are about 20 times more rods than there are cones in each eye. Another big difference between rods and cones is where they are located in the eyeball. So if I draw a very simplified diagram of an eyeball, and this is the optic nerve exiting the back of the eye. So this would be the front of the eyeball, this is the back of the eyeball. And as I mentioned in a previous video, the back of the eyeball is coated by a membrane known as the retina. So rods are actually found in the periphery of the eyeball. So they're found in this area over here and in this area over here. And there's actually a region of the retina right about here that actually dimples in, and this region is known as the fovea. And cones are mostly concentrated in this region in front of the fovea.
In the physics aspect of night vision, there are two different ways that it can work. The first way is called image enhancement and works by collecting the tiny amounts of light, including the lower portion of the infrared light spectrum that are present but may be imperceptible to our eyes, and amplifying it to the point that we can easily observe the image. The second way night vision is made possible is through thermal imaging. Thermal imaging operates by capturing the upper portion of the infrared light spectrum, which is emitted at heat by objects instead of simply reflected as light. Hotter objects such as warm bodies emit more of this light than cooler objects like trees or buildings. Image enhancement technology is what most people think of when you talk about night vision. Image enhancement systems are normally called night vision devices, or NVDs. NVDs rely on a special tube called an image intensifier tube to collect and amplify infrared and visible light. In conclusion, it can be said that night vision is a valid and possible superpower. There is multiple facts behind the development of night vision, and it's very possible that these optical enhancements could become a natural ability for the human species.